Thank you very much. I'm absolutely thrilled to have been asked to speak here today. It's absolute thrill. I've been reading Wired for quite some time now, so thank you so much. Now, in the program, it does say that I'm going to be talking about our handheld device for the purpose of diagnosing cancer. But I've kind of done the typical difficult speaker thing and changed that around a little bit. I thought it was more appropriate for me to talk about our program in infectious disease. And I think that will fit very well with what we've heard before. I've got a lot to get through, and I've been told I've got to stick to my 15 minutes, so I hope I'm able to make it, as you can see. Eek. So let's start with our story. Where have we come from? I'm sure most of you don't know too much about us and where we've come from and who we are and what we aim to do. We are a typical startup in the sense that we started in a garage. My garage in the south of England, not in a garage in Palo Alto. So this brings something new. And the way in which we started was novel as well. We're not a university spin-out. Our technology did not start there. Our technology started in here. And with my colleague Elaine, who sat there taking pictures of me and making funny faces, we put this together from scratch. And so that's something novel and new. This is an interesting picture. This encapsulates all my passions. As you can see, football and beer, dressing funny, genetics and inventing. And you'll see a lot of that as we go through this presentation. I have to apologize if I keep looking down. This is only the third time I've seen this presentation, as it was sent to me this morning at 2 o'clock in the morning. So we've moved from the garage, we gained some funding, moved to Cape Town for 2 years. And that's really where we honed our ideas. Really got to spend some time with the people who actually will benefit from a device like ours. I'm not really talking about the West and Western medicine. I'm talking about developing world, people who don't have access to molecular diagnostics and DNA sequencing like we do. So it was great to spend the 2 years down in Cape Town really learning and understanding their needs and what to do, and really focusing our vision. After 2 years, we moved back to the UK, and we've moved from London up to Newcastle in the northeast, which is, again, atypical, moving out of the Golden Triangle into the northeast, which has turned out to be very, very good for us and very good in terms of attracting government funds, which has been very useful. We have partners all over the world, which we will need, and that will become evident when you start to understand the vision as I move forward. So the problem, what is the problem that we face globally? In terms of infectious diseases, we have to worry about things like swine flu. So when you think about the onset of swine flu, it's truly remarkable. I mean, one of the things that strikes me about these images here is this suspected and confirmed cases after nearly 4 months are very, very small. So there's been 4 months in which this virus has taken hold and developed and spread through communities in different countries as well. This is alarming. For me, this is not good enough. This means that we could have contained this earlier, because look what happens in a very, very short time afterwards. You get this massive expansion until it hits the entire world. These countries that are not marked with red probably haven't reported their data, so this is another significant issue that we need to look at. It's not just new viruses emerging and spreading throughout the world that we have to worry about. It's viruses that we know about already that evolve, and they involve drug resistance. And this is becoming a significant issue. More and more we're seeing these problems. For instance, here, this is the spread of multi-drug resistant tuberculosis, MDR-TB, and XDR-TB, which is extensively drug resistant TB. And that means no anti-infectives will work, and this is obviously a massive concern for us. These things happen quickly. These things happen in an instant, within a hotspot, spread, proliferate, and then move out of that hotspot, as you can see here, especially around the Russian countries here. That's a massive concern. Now, we're tuberculosis, we understand a lot of the mechanisms of drug resistance already. There's a lot of money that's been put in to understand them, and that's helped us to track it. But things like malaria, for instance, we understand some of the mutations associated with drug resistance, but not all of them. We've got a hit list of about 20 mutations, maybe a few more that we want to keep an eye on, but we don't have enough data to allow us to really provide a clinical utility in terms of providing the genotype to a health worker on the front line. So in terms of the economic impact, as you can imagine, the impact of swine flu was truly substantial. We were thinking about in Mexico alone, you had a £55 million per day impact, actually reduced the economic activity by 1%. And then when you look at the market segments as well, including football, horrifically enough, a massive impact economically. And then moving into drug resistance, we see some alarming numbers here. In 2005, when we saw the original map, there wasn't a lot of red on it, but it's even then, £1.6 billion treating just MDR-TB as multi-drug resistant tuberculosis. This is truly alarming. It's something we need to get a handle on. And so, why is it costing so much? Well, one of the issues is when you're treating MDR, you have to use special anti-infectives. It costs a lot of money, obviously, to create drugs, but still, in terms of a pharmaceutical company, when you're looking at this market, it's quite small. So you're not necessarily motivated to keep producing anti-infectives because the market's not really there. And so per patient, you saw just $250,000 US dollars to treat a single patient in Tooting in St. George's Hospital. In the States, it's about 17,000 for a standard tuberculosis, 134,000 for multi-drug resistant tuberculosis. And when you get to extensively drug resistant tuberculosis, it's a significant investment to treat these patients. This is something that we really need to look at, we really need to get a handle on. And so our solution is to look not necessarily at the treating of the diseases, that's all well and good, but as we've seen, the problems with creating these therapeutics is they're expensive, and then to administer them and to ensure that we've got adherence, it's a big problem. So what we want to do is contain the issue. And we want to contain it through networking. So you've heard of, obviously, the internet. It's networked everything, provided us with a much better standard of life. We've heard of the Internet of Things, or as Cisco have told me outside, it's the Internet of everything, which is interesting because I don't think it really is the Internet of everything. So I'll see your Internet of everything, Cisco, and I'll raise you the Internet of life. And what do I mean by this? Well, I mean that we are going to interface the genomes of bacteria, of different pathogens, and we're going to translate the binary code, the genetic code into binary code. But that's not really enough. I mean, we obviously need to get that up to the cloud so we can start analyzing it. So when we're thinking about the developing world, we've got to think, how do we actually get that information up? So we've been partnering with satellite companies. We've been working with cellular companies as well, and also started to talk to some of these great wild ideas about sending up internet drones as well, so that we can see very soon that we're going to have coverage for the other 4 to 5 billion people in the world. And that really then allows the vision of the Internet of Life as we see it to come about. So that's one point. Having this device that allows us to sequence the DNA, that's great. So we can sequence the DNA, we can send it to the cloud. That gives us the GPS data as well, so we know if we do come up with a novel or new mutation or a hotspot for drug resistance, we'll see then that it happened at that point in time, which is great. But if you've only got 10 or 20 of these things in one country, that's not going to provide you with the coverage. So it's our idea that we're going to have lots of these things all over the world, and we're going to be able to identify any emerging threat, whether that's natural or biowarfare, and also whether or not we can pick up the drug resistance as it happens to allow us to contain it. The other aspect of that, obviously, is cost. So if we want lots and lots of these devices spread throughout the world, we have to make sure that the cost point is such that everybody can afford them. So we're now aiming for around about the £500. Truth be known, it's probably going to be a lot less than that. We've done a really good job in containing our costs within the device. In terms of the actual testing itself, that is dropping all the time as well. We're around about the $1 mark at the moment, and that will drop even further. And that's for a specific panel of mutations, not indiscriminate or de novo sequencing at this stage. So how have we got there? So we've been working in reinventing the technologies, reinventing the molecular diagnostic process, getting different devices looking at individual processes, either lysis, PCR, detection, sequencing, and then integrating them and getting them into a cassette. And then we've now got our first prototype. We have a prototype that's actually working, that's running a full sample to result DNA sequencing. It's running about 20 minutes right now, which is a little bit too slow for us, so we're going to be increasing that as well. And then soon after that, we'll be adding full DNA sequencing. And you can see just some of the pictures of the different technologies that we invented along the way to allow us to get to a point where we are now sequencing. How this works, well, you get a finger prick of blood, put it into the device. The device will then, it will blend it using beads, so break open the cells, releasing the DNA, and then we extract the DNA. We separate the sample, all the proteins and the lipids, and the DNA will just flow right through. This is a special filter that we've developed called the Q filter, and this separates this complex mixture of things very, very quickly, very, very rapidly, and we can get from sample in to PCRable DNA out in a little over 2 minutes, which is extremely fast. We then run through a continuous flow PCR, so we do 30 cycles in 4 minutes. So this multiplexes and amplifies up all of the genomic regions containing the mutations of interest. And then following that, we hybridize these amplicons, these specific regions of the genome that we've amplified, to a nanowire, as you can see here. And this hybridization event causes a change in resistance in the nanowire, which we can detect. So we're looking at a yes-no answer for certain mutations, whether or not they're present in that amplicon. Well, that's not enough, so we then have invented supercharged nucleotides. These consist of a standard nucleotide with a polyionic reporter moiety separated with a long linker. And so we can do a sequencing by synthesis reaction from that sequence-captured amplicon. And this provides us with more data, provides us with de novo data that we can determine whether or not we have a new species or a new mutation. All this is happening in 15 to 20 minutes. So unlike the standard process, which could take days in a standard Western lab, or up to months, as we've seen before with a new virus, we're actually doing it in minutes, very, very quickly at the patient's side, sending the data to the cloud and ensuring that the patient gets the right therapeutic at the right time. No more will that patient be running off and you will not be able to see them again. So where are we now? So we're now testing our device internally. At the end of this year, we will be moving our testing to the field in Gabon. We have a multi-site clinical trial platform set up with the government where we're not only looking at the performance of the device, but also looking at the performance of this real-time epidemiology idea that we call the Internet of Life. And all of this is being tested. We'll then replicate that throughout South Africa and then over in Asia as well. But for all this to happen, obviously, we need a lot of collaboration. We need people on the ground. So NGOs are very useful to us. We've been working with the Clinton Foundation. We've been working with FIND. They've got people on the ground helping us drive our vision and letting governments know that this is coming. We're also working with CDC and other epidemiology outfits. We've been actively recruiting people who want to help. I mean, this is not just about us. We're creating the device, but we want other people to come into this and help us. In terms of network architecture and data security and data sharing, we're working with some very large US companies on that. Not Cisco, so if anyone's watching from Cisco, I'd love to talk to you. And then obviously the security protocols. And then even more important is the government. Data sharing is going to be very, very important for us. And obviously the health workers. So to wrap up, I mean, the big overarching theme here in terms of the Internet of Life is that we will create a bio API, a way of interfacing the DNA with the binary code and shoot that into the cloud. We'll be doing this in real time. We'll be doing this all over the world. And so that any mutations that occur or any new viruses that occur, we will then be able to pick up. And when we pick these up, we'll be able to contain it immediately by sending the right drugs, the right resources, and the right people to contain it. And that's it. Thank you very much.
So I guess, is it too early for you to have one in your pocket?
No.
How long?
But I'm not allowed to show it.
So you have a prototype?
Yes.
How long until you're able to pass it around? How far away are we until it's...
And so if anybody wants to come and play with it, sometime after the summer, we will let you hit the streets of Newcastle and go and test people, just to demonstrate how easy it is to use.
Great. Best of luck. Thank you, Jonathan.
Thank you very much.